Good evening, ladies and gentlemen. Welcome to the first in the winter series of lectures. I'm afraid Dr. Jim Wilson has been delayed. He missed a connection from Luton to Glasgow, but rest assured he is on the current flight and will be arriving in Stornoway at 6 o'clock. We have our Vice Chairman, Mr. Angus McCormack, or Speedy Gonzalez, who's going to take him up to the chamber for us. Tonight, the speaker is going to talk about Viking heritage and the genes, not genealogy, genes. And this is it's a carry-on from what he has done before in Orkney and Shetland. And one of the benefits that we get from finding out whether we're Picts or Vikings or Anglos or Irish is that in small communities we do get there is a feedback from our genes into whether we carry genes that affect our health, and from that the benefits can be accrued. Now, Dr. Jim Wilson will tell you all about that. Sorry, it should have been Professor Jim Wilson will tell you all about that when he arrives.
So, without further ado, I'm Jim Wilson, and I'm the leader of the Viking Gene study, and I've come here to tell you a little bit about it today. So I'm going to explain to you why I'm interested in studying the Northern Isles and the Western Isles, and introduce to you the idea of a gene pool, a distinct gene pool. And then I'm going to tell you why this is important, the idea of using DNA in medicine, something that's just beginning to happen and we're really on the cusp of. I'm going to give you a little bit of an update about the Hebridean side of this story, and close with a little survey we've recently done of our participants to find out what they want us to do, which is not quite always the same thing as what I want to do. So everybody always asks me, I work at the University of Edinburgh, why don't we study the good Edinburghers, the people of Edinburgh? Why do persist in studying these faraway places? And it's because they're isolated. I don't need to explain to you. My trying to get here is evidence of the isolation of this place. But it's not so much about the isolation today as the isolation in the past. And this means that there's a limited number of founders. If we think of a population being started at some point in the past, how many people came to begin the population? I'll talk a little bit more about this in the future, in a couple of minutes. And another thing that you'll be aware of, those of you studying genealogy, is that marriage, in the past at least, tended to occur within the islands. And these are two sides of the same coin. If you have a limited number of founders, then marriage is going to occur within the community if there's not a large amount of immigration. And this is true from Eriskay to Orkney, from Yell to Islay. And what happens in this situation is that the people who live in this population begin to develop a slightly different gene pool, a slightly different group of genetic variants. And again, we'll go through an example of this later. What happens is that many genetic variants that I would find if I studied the people of Edinburgh, we wouldn't find here. They're lost. They were never brought here. Or if they were brought here, they died out. But the other side of the coin is that other variants get more and more common, far more common in an island than they might be in London or in Edinburgh or Glasgow. And we call this genetic drift. The variant drifts up, or it drifts down and drifts away. So the studies I'm going to tell you about initially are the Orcades and Viking studies. These are the first studies I set up. Orcades, the Orkney Complex Disease Study in Orkney, where I come from. And Viking, the Viking Health Study Shetland, in Shetland, where my dad's from. So I won't need to lecture this audience on why these populations are slightly different. Of course, they belonged to Scandinavia, to Norway and Denmark until 1468, not all that long ago, really, in time. And they had been settled by Scandinavians over 1,000 years ago, actually on the way to Lewis here and round the so-called Sea Road to the Isle of Man and onwards to Dublin. So these populations are part Scottish and they're part Norse. They're hybrid populations, which makes them slightly different from other populations. Now this is the first genetic result that I'm going to show you. Don't be overwhelmed by it. It's a very simple graph. So every dot is an individual human. It's a representation of their genome, a representation of their DNA. And the closer together the dots are, the more closely related they are, the more DNA they share. So if you see the blue dots down at the bottom left there, these are people from Tuscany. And you can see if I take one Tuscan, he's most closely related to some other Tuscans. The Tuscans are each other's closer relatives than they are to the Finns or to the British. But then you see the Spanish here in green, and again the same thing's true. Any given Spaniard is more closely related to other Spaniards than he is to British or to Finns. But the Spaniards are closer to Tuscany than they are to the other European populations. So we're seeing here a Tuscan gene pool in central Italy, a Spanish gene pool, but because Spain and Tuscany are near each other, there's also a Mediterranean gene pool at a higher level. Finland's away over here on the side, but if we focus now on the British, and actually these black dots are Americans from Utah, we see that they're very similar to each other, the British and the Americans. But Orkney in light blue and Shetland in purple at the top are distinct genetically, even from the British. And what's more, they are distinct at a European scale. They are as distinct in some ways as Finland is, another country far away. So it's really this genetic distinction that I want to study and that I think is important. It's because of this deep shared ancestry over centuries from a small number of founders that creates this drift. The populations have drifted away from the general British population. Now I'm going to illustrate to you now the so-called genetic structure across Scotland. So how ancestry varies across Scotland, which people are related to which other people, where are these different genetic groups found, what are the patterns of the genes as we look across geography, and finally which contributions from different source populations can we find. So we performed a large study using a large number of different cohorts, looking across the whole of the British Isles and Ireland. We then put it through a complex computer program to understand how these populations related to one another, just like a family tree. But instead of being a family tree with individuals at the end, this is a family tree of populations. The closer together the two populations are, the more closely related they are. And the further apart, the further apart. So you see immediately that there's two huge big branches to this tree, in blue there and in red. And the one branch is actually all the Orkney populations and the Shetland populations. They're the most genetically distinct groups in the whole of Britain and Ireland. Over on the other side, here we have England, all the different populations of England in this branch, who are most closely related to the Welsh branch, which might not be that surprising given their geographic relationship. Here, in the middle, we have an Irish branch, then a Hebridean branch in between Ireland and the Scottish branch here. But within Scotland, we can further break things up. We have a northern and eastern Scottish branch, and then we have a southern and western Scottish branch, two very large divisions of the Scottish mainland in terms of genetics. And further, within the southern and western branch, we find the people of Ulster, due to the plantation of Ulster, and the people from the Isle of Man, both of whom are actually genetically Scottish in our analysis. So this is the clustering of British and Irish DNA. But we can go a stage further and look on a map. So here again, each dot is one individual, and they've been coloured in according to their genetic cluster. So the computer program, I should remind you, had no idea where these people came from. This clustering is based on DNA alone. We then mapped it back to the map by taking the midpoint of the birthplaces of the 4 grandparents of the person who gave us the DNA sample. So the first thing I want to point out is this big blue group. So all across the plains of central and southern England, everybody is genetically indistinguishable. They all belong to this large English group. It means there has been movement across here, probably in the Industrial Revolution, or maybe since, and there's not been time for genetic drift to take effect and develop a different gene pool. When you go to the very extreme, here in Cornwall, they have a Cornish gene pool, as they do in Devon and the Welsh Marches and the north of England. And you can see similar things across Ireland, different colours there, different genetic groups. But in Scotland, it's the most diverse place of all. Many, many different colours, many, many different genetic groupings. And I want to highlight just a few to you. Here we have a red gene pool, the Aberdonian gene pool, and we can tell using DNA alone Aberdonians from Dundonians, even though it's only a few tens of miles apart. And they're large cities, not rural parishes or islands. So the people of Aberdeen have been marrying people from Aberdeen for centuries, and the people from Dundee have been keeping with people from Dundee in order for us to find this pattern. Now, so there's an Aberdonian gene pool and a Dundonian gene pool and all these other places here. Now zooming in on the populations that I've studied the most, Orkney and Shetland, we can see incredibly fine detail. Each of these different isles and some of the parishes that are connected on the mainlands of Orkney and Shetland, we can tell apart genetically. They have their own gene pool. And the most extreme case is here in Burgh Isle in Shetland. So Burgh Isle is 1 mile off the coast across Clift Sund from Quarf in Shetland, and we can tell people apart who come from 1 mile apart. This is how tight they have kept their genealogy. We see exactly the same thing in Orkney in the Isle of Rousey, which is again 1 mile across Einhallow from Evie, and we can tell them apart. The Isles of Westry, Sandy, Stronsey, Southronaldsey, Yell, Unst, Whalza, all these places we can tell apart. Now, to a different kind of analysis. Rather than looking across geography and seeing how people relate in trees, we're asking, if we take the genomes of these British and Irish people and model them as being made up of the genomes of continental European people, what do they look like? Do they look similar? Do they look different? And which continental populations do they come from? Do they come from Italy, France, Belgium, Germany, Norway, Sweden, etc.? So we put it through this model, and the first I want to point out to you that's so obvious is this huge red French-like ancestry. Everyone in the British Isles is coming out with quite a large proportion of this, which we believe is a sort of Celtic layer, one of the older layers in our ancestry. It's found everywhere, but it's most common in Ireland. Then we have an orange grouping, again found all over the place in variable amounts, and this is a German-like ancestry. The genomes are being modelled as matching Germans. And this can be explained most obviously in England, where it's the most common, as Anglo-Saxon ancestry. But if you are involved in deep, deep genealogy, and you look into the genealogy of any Norman family, you will end up with people called Franks, which France is named after, but who are a German-speaking people, and will come out in this analysis as German. So the Norman contribution to Scotland is probably shining up here as German-like ancestry, and there will be other ways, no doubt. And finally, in blue shades, we have Scandinavian ancestry, the royal dark blue one here being Norwegian. And this is highest in the Northern Isles, but is also present more widely across Britain and Ireland. So we wanted to zoom into this in a little bit more detail and do a simpler model just using England, Wales, Scotland, Denmark, Sweden and Norway for Orkney and Shetland, because I was interested where in the British Isles has the most Scandinavian DNA of all. And the answer is the Isle of Yell in the north of Shetland, coming out at about 28% Scandinavian. And you see that all of these places have a decent amount, between a fifth and a quarter Scandinavian, but it varies. So central Shetland there, so Tingwall is actually half that in Yell. So it's still showing geographic variability, and it would be very interesting to be able to draw these graphs for parts of the Hebrides. So this might explain some of the activities in Orkney. When you are having your Christmas lunch, the men of Kirkwall are out having a brawl. Now it's a game of street rugby called the Ba, involving scrums of up to 150 people, with one of the goals being the sea. You can see the steam coming off the pack here on Christmas Day, while more famously in Shetland a month later, having spent an entire year building a Viking longship, which they call a galley, they then burn it to cinders. So I now want to have you think a little bit about the idea of a population bottleneck and how we get these gene pools. And it relates again to this idea of how many people live in Lewis, how many people live in Orkney, how many people live in Shetland. So these days in Orkney and Shetland it's about 21,000 in the one, 23,000 in the other. And how many lived there in the past? That's actually more important for our DNA. And actually in Orkney it hasn't fluctuated that much. It's been between about 10,000 and 25,000 for most of the last 1,000 years, which is essentially not changing very much in size because so many people went off to the colonies. Otherwise there should be hundreds of thousands of people in Orkney and Shetland. If you had 10,000 people in the past, where have they all gone? So that's giving us a bit of an idea, but I don't think Orkney was founded by 10,000 people. And we don't know. Without a time machine, I'll never know how many people founded Orkney, but we can get some clues. So there are surnames, very, very useful in Orkney. There are 260 surnames that are Orcadian. Some of them are indigenous, only found there, and others of them are shared with Scotland, but are much, much more common there. So that's 260, in the absence of illegitimacy, that's 260 men, at least, that founded the population, presumably with 260 women as well. So there's a very bare minimum. We can also look to Iceland, where they have wonderful records, including one called the Landnáma Book, the Book of the Taking of Land, which lists 3,000 mainly men, mainly from Norway, who took the land of Iceland. There was no native population there. And if you look at Iceland, it's massive compared to Lewis or compared to Orkney. So I think there would have been many fewer than 3,000 in Orkney, more than the low hundreds, less than the mid-thousands, maybe a couple of thousand. But in any case, many, many fewer than in a cosmopolitan population. How many people founded Glasgow? I mean, what does it even mean, you might say to me, but think of all the people who moved to Glasgow through the centuries. Thousands and thousands and thousands upon thousands. And this is why we see this difference. And we call it a bottleneck because of the idea that in the bottle is the original population, and you've got different marbles or DNA types, red, green, orange. And as you go through this bottleneck where only a few people move to the island, not every colour arrives. Not every marble comes through the bottleneck and not every DNA type gets there. In the new population, you see there's no red marbles. This is a DNA variant that didn't make it, and the frequencies can change. And this happens again and again and again. I'm making the argument that it happened when the Norse really set up the present-day populations of these islands. But the Black Death had an enormous effect, at least on the Northern Isles in 1349. There were smallpox epidemics all over the place. There was a famine in Orkney and likely here. 3,000 people in Orkney died between 1620 and 1640. That's again going to have reshaped the gene pool of the islands. So I'm now going to go on to report a little analysis we did in Shetland. We were lucky to raise funding through the Scottish Genomes Partnership so that we could read all of the DNA of 500 of our volunteers from Shetland, so whole genome sequencing. And we had samples from Lothian, from Edinburgh, for comparison, where we'd done exactly the same thing. And I knew they'd be different. That's why I set up these studies. But I was very surprised to see that fully 10% of all the variants we saw were either specific to Shetland or were 10 times more common there than they were even in Edinburgh. And this is not a small number of variants. This is 400,000 DNA variants that were Shetland specific or vastly enriched there. So it was much, much more different than I had imagined. Of course, we all share 99.whatever% of our DNA, but because Malcolm and I differ at 4 million letters of the DNA code, even a small percentage can be a large number of letters. And given that one DNA letter can cause disease, there's obviously scope here for something important. And this is the idea of a gene pool. This is a cartoon, I think it's of me, from the Shetland Times, pointing out, as I always do, how important the gene pools are. And it comes from this deep shared ancestry. And this got me to thinking that if the fruits of genomic medicine are to be shared equitably across Scotland and elsewhere, then we need to understand these variants that only we have. And no one else is going to do it for us. And so that's why I'm here, and that's why I do what I do. So, I hope I've persuaded you there are subtle differences in the DNA of the islanders. But Does it have any meaning apart from being really interesting in terms of history? I would say yes, and I'm going to give you a couple of examples. Because 10% of the variants are different, it means a proportion of the medically important variants are also different. You must understand most of these variants are doing nothing. They're just an error in the DNA, they don't matter. But a small number of them probably do matter really quite a lot. And I'm going to tell you now about an example from the Viking study from Shetland. So during the recruitment, a volunteer came and shared a letter that she had from the NHS, and it told her she had a relative with long QT syndrome, which I'll explain to you in a second. And it was an invite for her to be screened. This is how the NHS do it. If your brother has a disease, they cascade out to you, to the nephews, the nieces, and they get tested, and then they follow it like this, one step at a time through a family, cascading out. So, long QT syndrome, you will know of it. If you think, you hear these terrible stories sometimes, 25-year-old lad, footballer, running around playing football, drops down dead like this. That's usually because he had a pre-existing heart condition, and the exercise exacerbated it, usually because he had long QT syndrome. And this runs in families. It's called this because of the QT interval. So here is an ECG trace, a trace of the electrical activity of the heart, 2 heartbeats, showing here, and there's an interval between the Q and the T that's called the QT interval. And this is a healthy person, and here's someone with long QT syndrome. So it's just elongated a little bit. It doesn't look like much, but you have to remember that the heartbeat is one of the most carefully timed things in our body, and this slight disruption can trigger dangerous arrhythmias, including the most dangerous one of all, fatal cardiac arrest, when the heart stops. And it's usually in response to exercise or stress or surprise. But it's not the end of the world. There are treatments, there are drugs. You can avoid the triggers if you know what they are. And at the end of the day, if necessary, pacemakers can be implanted or implantable defibrillators. So this is a disease that we think of as a simple disease. It comes from one gene, in this case the gene KCNH2, which encodes a channel involved in relating the electrical signal of the heart. Now, it's very, very rare in the British population. There's a large database called UK Biobank, and it's less than 1 in 100,000 there, so exceedingly rare. We found one in our 500 Viking samples. It wasn't the lady with the letter, she was fine. Someone else carried this variant. And this person actually had 42 relatives in our study, but they weren't in the guys that had their whole genomes done. So we looked for them individually, and we found 2 others. So here's an actual DNA trace. You can see how people are the same. Person on the top, person on the bottom, they're matching. C, A, C, G, C, A, T, C. And then at this G, the bottom guy has a G letter of the DNA code. The top guy has an A and a G. This is the mutation, this is the genetic variant that causes long QT syndrome. And here's the family tree, this time the family tree type that you're all used to. Here with the arrow on the left is the man who we first discovered, had the variant, and then his mother's sister's daughter and her daughter in black also carry it, and we can also infer that the deceased mothers, who are not in my study, also must have carried this variant in order for it to have come down this tree. We also see in green other people, close relatives, brothers and sisters and nephews who are in the study but do not have the variant. They had the good luck of the genetic lottery not to inherit this variant in the family. Now, we were able to take these 3 people, and because we had a lot of genetic information on them, we could define what's called a haplotype. It's like a barcode of DNA. And here are the barcodes for this part of the genome for these people. And we all know that we get half our DNA from mum and half our DNA from dad. And you can see here we have on the one side from mum, from dad, from mum, from dad, and from mum, from dad. I think if you look, you'll agree with me that the ones I've arrowed here are identical. In fact, I can pull them out, and you see they're exactly the same. So proving that they come from the one event, the one piece of DNA, the one mutation. It also means we have the digital track of it now, so we can go out to all our samples and look for it. We have 20,000 samples from mainland Scotland. We didn't find it at all. But when we looked in the rest of the 2,000 Viking samples, we found another family, 2 more people. and they're not related to the first family. We trace them back to about 1770, and so they could never have been found by cascade screening, going from brother to nephew, and so on like this. So they're either very distantly related, you know, sharing an ancestor in 1700, or there has been what we call an ancestral non-paternal event, and they actually are related, but they don't know that they are. So this is a way that the cohort studies like this can help, because although this variant was known about by NHS Grampian Clinical Genetics, these people were not known about, and we've been able to feed this information back to them. So this is the beginning of the idea of genomic medicine, and particularly how cohort studies such as Viking Genes can help with this. So genomic medicine is just a fancy word for using DNA information as part of your medical care. But what we can contribute to this is a number of things. One, just like this family B here, we can identify families that could never have been found. But the other thing that we can do is get a much more accurate idea of the effect of these variants, because we're getting essentially random individuals to come to us, and then they're being tested. If you work in a cancer clinic and you find a mutation in the people with cancer, of course you're going to think it's related to cancer, because you only look at people with cancer. I can then go into samples of thousands of people who came to me nothing to do with cancer at all, and ask truly how many who have this have cancer or not, which is powerful and important information. Professionals should take them into account when they're assessing the picture of a given individual. And this is one of them for Shetlanders at least, because it's 150 times more common in Shetland than it is elsewhere in Europe. And this is really what got me going to find out what other harmful DNA variants are common in the populations that I study. And it's very important for populations like ours. Also, other isolates, Isle of Man, Irish travellers, famously Ashkenazi Jews, and so on. Much more important than in cosmopolitan populations who are being studied anyway. So there's a little coda to this story, because ID4 from Family B actually got in touch with us in response to a social media promotion of the new study, Viking 2. And she said, "I have lived in Aberdeenshire for over 30 years. Both sides of my family are from Shetland for as far back as we can trace. I thought it would be interesting to take part, predominantly as it looked like a free medical. In those days, we went to the islands with nurses, they got their blood pressure and their cholesterol measured and got a sort of free medical. I received a great report on my overall health, and I filed it away at the time thinking I would never see it again. However, several years later, I was contacted by my GP as they had received notification of a rare genetic mutation via the Viking Health study. It was discovered I had long QT. Through genetic testing, we have since discovered my father and my 9-year-old daughter both have the same genetic mutation. Both myself and my daughter now take daily beta blockers to control our heart rhythm and see a cardiologist every 6 to 12 months. Neither of us have ever had any symptoms and live very fit and healthy lives, and we will never be certain, but it is possible that taking part in this survey has saved our lives. Thankfully, with the medication we are on, we are both able to continue to do all the things we love, including running. I can't recommend this survey enough. You just never know what you might find out." So I've spent the last 20 plus years of my life working in genetics to try to understand the biology of disease so that drugs can be designed, better drugs in the future. So I have to go home at night thinking I'm helping my children's generation and maybe future grandchildren's generation. Here, it was very moving for us, for the team, because we had directly affected someone's life just like that. And she was so kind to come forward, Ingrid, and tell us the story and allow us to put it on our website. So I'm now going to tell you about another variant, this time from Orkney, which we discovered through our sort of broader survey. And this is a more famous variant. You may have heard of the BRCA1 and BRCA2 genes that confer a risk of hereditary breast and ovarian cancer. The most famous carrier is probably Angelina Jolie. We found 20 carriers in orchids in the Orkney study, which is huge, and so much so that it was actually already known to clinical genetics in Aberdeen, but it was 480 times more common in Orkney than it is in England. This is genetic drift, but not the kind you want. I was very interested in where this actually came from. So we have grandparental information for all of our participants, including the carriers, and you can immediately see here there's a very strong overrepresentation of grandparents from the Isle of Westray in Orkney. And indeed, a little bit of genealogy showed that we could link everybody back to Westray ancestors, but not to the same couple, unlike in the Long QT family. Back to 1750, 1720 even in some instances, and they just refused to link up, but they're all in Westray. So I interpret this as being the mutation occurred in like 1650, and these people do link, but I just don't have, there is no data. We don't have the information. They're bound to link. It's just an older mutation. And in fact, when we did the digital barcoding trick, the haplotypes again, it's hard to see, but there's a very slim blue line goes right down the middle of all that, showing that they all share the exact same piece of DNA, proving they all descend from the same person at some point in the past. And we actually found 4 of them in the large UK Biobank study of 500,000, and some in a project from Nevada in the States, and they had the same haplotype. It means their ancestors are from Westray. They might not know it. It might be their great-grandmother or their great-great-grandmother, but they are Westray descendants, and they carry the Westray BRCA mutation. So laboratory studies have shown that this is a harmful variant. And NHS Grampian, as I said, have known about it for a while. They had a total of 14 female carriers and 6 obligate carriers. So you remember in that first family tree, the son's mother's sister's daughter, and these two I coloured in grey, they're obligate carriers. They're not in my study, but I know for a fact that they carry the variant. So they're sometimes useful to see what's going on. And amongst these women, they had 9 ovarian cancer cases and 5 breast cancer cases, and 1 person who had both. Now in our study we have 7 carriers and 9 obligate carriers. We only have 1 ovarian cancer case and 2 breast cancer cases, which seems a little bit different. Partly it's because some of our people are younger. They haven't yet come to the age where these cancers mostly happen. But it might also partly be because the true nature of this mutation is it's not quite as bad as the clinical geneticists first thought. It is possible to live to an old age with this variant. One of our carriers is dead now, but she reached the age of 90, which is not bad going. But there's still work to be done to understand how likely are you to get the disease if you have the variant. Does it double your risk? Does it increase your risk by 50%? There's no one-to-one correspondence. If you have it, it doesn't mean you're going to get cancer. And to be honest with you, I don't know if we have enough humans carrying the variant to know the answer to this. But there's certainly one thing that's true. If you carry the variant, you should be screened far more often. Just be screened, and then you can pick it up if it does happen. It allows you to focus healthcare on who needs it most, long before we get to the stage of mastectomies and oophorectomies, so removal of the ovaries. So these 2 variants I've told you about today are examples of what we call actionable variants. They're harmful variants that are linked to serious disease, but which we can do something about. We can take action, hopefully to prevent the disease or at least to improve the outcome. In each case, early knowledge of the risk could enhance how well the preventative measure or the cure works. So it's beneficial to know, and we estimate that between 1 in 50 and 1 in 100 volunteers actually have an actionable finding. They could be long QT, they could be hereditary breast or ovarian cancer syndrome, there are hereditary bowel cancer syndromes, familial high cholesterol, much more common in the Hebrides, for example, and a variety of rarer metabolic and other diseases. This would work in the future. We hope that people that we highlighted would provide a repeat DNA sample for NHS clinical genetics to validate, and they'd follow the pathway of care of the NHS. And we hope to begin doing this work next year. So in the Viking 2 study that some of you might belong to, you will have consented to the return of results by ticking the yes option here in this part of the consent form. And I'm delighted that in a snapshot from some time ago, 98% of volunteers said they did want to know, while 2% exercised their right not to know, and that's up to them. But they're given the option to know or not to know. The trouble I have with this is that the Orkney and Shetland cohorts, we have the DNA data available, but we don't have the permission. for Viking 2 and Viking genes, we have the permissions, but we don't have the DNA. So I'm hoping by 2023 to get the permissions from the old lot and to get the DNA from the new lot, and then we can actually do this properly. So this is a survey in Orkney and Shetland of the actionable variants of 73 genes that are the American College of Medical Genetics has decided have actionable variants in them. And we found that 1 in 66 people carried an actionable variant. Mostly cancer risk variants, but also some cardiac risk variants and an assortment of others. So we have a lot of work on our hands to understand these and work through them. So bringing this more up to date, we launched Viking 2 in January 2020, just ahead of COVID. We wanted to double our volunteer numbers from 4,000 to 8,000 to help understand these rare variants. When things are rare, you need very large numbers to find enough of them. And again, people from the Northern Isles, but this time not involved in clinics in Orkney and Shetland, but an online survey. So people could come from anywhere in the world as long as they had at least 2 grandparents from Orkney or Shetland. And the DNA would be provided by saliva rather than by blood. We've had a huge response, as you can see here, people from all over the Northern Isles, but also the Diaspora. Here are people across the towns and cities of Scotland who have Northern Isles ancestry. And indeed diasporas I hardly knew about, extending down across England, although very, very few in Ireland. And actually across the world in the former colonies, but some in very unusual places in Hong Kong, in Anchorage, Alaska, Honolulu, Hawaii. The people of the Northern Isles have gotten to most of the corners of the earth. Now bringing things more up to date, I've been interested in the Hebrides for many years, as you'll see, and I wanted to expand the study to allow people in the Hebrides to join into this work, and also to allow me to do further population genetics. As we've seen in our tree at the beginning of the lecture, the Hebrides also have features of isolated populations. They formed their own branch. These were people principally from Skye and from here in Lewis and Harris. We also had populations from Islay, which also branched to themselves, but this time with others in Argyll, rather than with those in Lewis, Harris and Skye. So we opened up Viking genes to the inner and outer Hebrides this year in June, and we've had a wonderful response. And what excites me most is actually all these places that have never had any DNA work at all. Mull, Tiree, Barra, the US Benbecula, they're almost entirely unstudied, and so we don't know what we'll find in these places. I started life as a population geneticist interested in ancestry, and so it excites me to see how much Irish, how much Pictish, how much Scandinavian we're going to have, all the way from the Rins of Islay to Ness in Lewis, and from Hjort to Broadford in Skye, for instance, all across these places, if we can recruit sufficient people from each place to represent the ancestral gene pools in these places. Now, the previous studies of the Hebrides are few and far between, but I happen to have been involved in them. Just after my PhD, you might remember the Blood of the Viking study, a BBC One or BBC Two maybe, series featuring Julian Richards. So there were actually 59 Lewis men and 29 from Harris in this study, because the grid that we'd placed across the British Isles, one of the corners was not far from here in Stornoway. And then I was involved later in a BBC Alba series called Eirachuan, or On the Ocean, where we recruited 123 men from Lewis and Harris, Skye, Islay, and also a sample from the Moray coast in eastern Scotland as a comparison to see if the Hebrideans hung together and differed from the people in Moray. So I'm going to show you a quick outline of what we found, categorized by different lineages. So first, looking at the Irish side, you can see here pie charts of male lineages, Y chromosomes, for a variety of places, and you can see quite a consistent Irish contribution to Scottish DNA. To Lewis and Harris, quite high, 2 different kinds of Irish DNA, Skye, Islay, Caithness and Moray, but funnily none to be found in Orkney. And with much larger samples since, we have found a very, very small number of Irish lineages in Orkney, but they probably went from Ireland to mainland Scotland, and then a Scot went and moved to Orkney. Then the lineages everyone's obsessed with and wants to descend from, the Norse male lineages. Here we have, again, high representation in Lewis and Harris, but also even way down in Islay, we have quite strong representation of Norse lineages, less so in Skye, rising up again in Orkney. Shetland is actually even higher, although it's not here. We have reasonable representation in Caithness, and this time it's Moray on the east coast that has none whatsoever. I think if we had much larger samples in Moray, we'd find a few, but you're not going to change it up to, you're nearly at a quarter here, whereas in Moray, maybe it's 1 or 2%, I don't know, but it's nothing like a quarter. And then we have Pictish male lineages. These are a sort of a favourite of mine, actually, because everyone forgets about the Picts and they get ignored. Whereas I think we have a very large, as Scots, have perhaps the majority of our ancestry actually comes from Picts. It's not so easy to tell a Pict from an Irish lineage, but there are a few Pictish lineages that I'm 100% certain of, and I have put them into pie charts here. And the thing that I think's most noticeable here is just how Pictish the island of Skye is. We have Pictish lineages everywhere, Moray, Orkney, Caithness, but Lewis and Harris has a good few, and Skye has really, really quite a lot. This is a bare minimum. There will no doubt be other Pictish lineages, and at some point when I'm not so busy, I may look at this again and see what else we can do to update this picture. And having lots of new samples in Viking genes, of course, would help that work. So that was from Eirachuan. Now looking back to Blood of the Vikings. Now remember, this is from around about 2001, when DNA testing wasn't anything like as detailed as it is today. But what was useful here is that the Lewis and Harris samples were separate, whereas in Eirachuan they'd gotten mixed together, and I couldn't tell exactly who was from Lewis and who was from Harris. But you see these pie charts here, we're seeing higher Norse in Lewis, 19% versus 11% in Harris, whereas it's the other way around for Pictish. Higher Pictish, 17% in Harris, 10% in Lewis. So Harris is actually looking quite like Skye to me. Lower Norse, higher Pictish. I also found that in Blood of the Vikings they had a tiny sample of Barich, which showed that a huge number of them belong to this S68 group, which I interpret as being Pictish. And this gets controversial for some people here, but this is because most of the men called MacLeod and most of the men called MacNeil actually have Pictish Y chromosomes. Even if their grandfathers and great-grandfathers told them different stories, I'm afraid that is the case. And it's a very cool Pictish Y chromosome because it's found in Orkney and it's found in the Hebrides and it's basically found nowhere else. So it's not your Moray, Angus and Aberdeenshire Picts. It's the people who lived on the Isles. Hopefully ancient DNA will come to my rescue and we'll learn more about these things in the future. So, bringing this right up to date towards the end of the talk, we have Viking 3. I've been on a tour from the beginning of summer of 17 Hebridean islands promoting the study, and I started actually in the Western Isles in June, and we've had an amazing response. Here's an updated graph of where people come from, and we've had over 1,000 DNAs back already from people who have joined the study, so I'm completely delighted, but I'm still looking for another 1,000 to bring the total over the whole study to 10,000 before the funding runs out early next year. I'm going to Islay and the Southern Hebrides. Well, I'm going to Oban tomorrow, and from there on to visit some of the Southern Hebrides to continue spreading the word. But hopefully you can help me as well by encouraging any people who have not yet joined, who have at least 2 Hebridean grandparents, to volunteer. It doesn't matter where in the world they live. It doesn't matter if they have no relatives in the study or all their relatives are in the study. We want as many as possible. And also women have been much better at coming forward than men. 65% women, I believe. So we need more men to come forward. David, who works for me, has created another amazing graph I'll show you here, where we can learn a little bit that I think is interesting about the diasporas from these various places. So we're going to show you some maps. They're coloured in depending on where their grandparents come from, not where they come from. So Orkney, yellow; Shetland, orange; not anywhere in the Northern Isles, red; Western Isles, by which he means all of the Hebrides, and some people have ancestry from Orkney, Shetland and the Hebrides who are in green. So we see Shetlanders in Shetland, and Orcadians in Orkney, and also Hebrideans in the Hebrides. But look at the mainland of Scotland. It's quite interesting how you see on the west coast of the mainland of Scotland many people with Hebridean grandparents who've joined my study, and on the east coast predominantly people with Orkney and Shetland ancestry, particularly around Aberdeenshire. But if you zoom into the central belt of Scotland, you see this huge component of Hebrideans in Glasgow. So this plot has, let me think, 4,000 people with Orkney and Shetland ancestry in it, and 1,000 people with Hebridean ancestry. So there's actually for the blue Hebrideans to be showing up in Glasgow so much, there's actually far more of them because it's weighted against them, because we haven't recruited as many yet, whereas it's more mixed in Edinburgh. And when we look across the world, you can see that the people in the prairie are Orcadians who went to farm the prairies of Canada in Manitoba, Saskatchewan and Alberta. And in New Zealand, most of our volunteers come from Shetland, and indeed one of the Prime Ministers of New Zealand, a distant cousin of mine, actually came from Shetland. So we're beginning to see these patterns out with the islands of Scotland. So I'm going to bring this to a close now by telling you a couple of things about our website. If you're interested in this kind of work, we publish many papers, and I'm not suggesting you start reading scientific papers, but we put lay summaries on our website. Here's a bunch of papers, here's another bunch of papers. Many different studies come out of this work because we looked at so many different aspects of it, and I'm very interested in many different things. So go to ed.ac.uk/Viking, and there's lots of material there. And the one thing I'm going to highlight to you is our relatively new pages on engagement with the public and involvement of the public. And the most recent aspect of this is a survey that we did. We had 1,400 of our volunteers fill in a survey on what mattered to them. So we first asked them, which diseases should we study? And I was quite surprised actually, because the top was Alzheimer's and dementia, something we really haven't studied and is quite difficult to study. But this was tied pretty much with heart disease, which is very important to the participants, and in third place was stroke. And the disease they were least interested in us studying was COVID-19, probably because everyone's quite sick of it. 3% of people thought we should be studying that. I then went on to ask them which of a number of sort of research areas they supported, things that we do, things that we have the skills to do. And the top three were understanding the genetic distinctiveness of the Northern Isles and of the Hebrides, and I think I spend a lot of my life doing that. The next thing was to study the risk factors for disease, so that's studying cholesterol, blood sugar, blood pressure, not diseases themselves, but the risk factors that cause disease, which again we spent most of the last decade doing. And the last thing they were very supportive of was work on actionable genetic variants, these kind of variants that you ought to know about, that we're trying to work on with the hope of returning them to people in due course when we have all the permissions in order. We also asked them a question about genetic history, and actually beat everything. 79% thought we should use our time and our money to learn about the genetic history of Scotland using our data. So, all that remains for me to do is to continue to beg you to please sign up, spread the word to friends and family at home and abroad. If you are one of these people who has signed up but then done nothing, you can go in and complete the questionnaire. If you've completed the questionnaire but you haven't returned your spit kit, it's not much use to me without the saliva, so please do that. Yeah, please spread the word, and hopefully I will come back to tell you more about the results of it in due course. It only remains for me to acknowledge so many people over so many years, funding bodies, individuals, colleagues, particularly Rachel, David and Shona, who you see here in the picture, who are the Viking Genes team dealing with participants, but most of all to all the Viking Genes volunteers all across the world, in Orkney, in Shetland and now in the Hebrides, without whom we could do none of this work. Thank you very much.
Jim, I'd like to thank you very much for coming along. You've flown in at late notice. And in reality, we've got to say, if people do participate, we all are going to benefit from it. That's the crux. Even if men have to be hypnotised.
Yeah, my job is to provide an evidence base for the NHS
Yeah.
to do the best that they can. I'm not in the NHS, but if we don't do that, your London ain't going to do it for you. So our populations are different, and so we should learn what we should do about, if there's something we can do about that, and that's it.
Thank you very much, and encourage your neighbours, your family to participate. And you know, it's not just Professor Jim Wilson, it's the community. With that, I must thank you very much, and thank you. Good night. And the next lecture will be by Ken Galloway at the end of October. Yeah. So good night.
Thank you.